Alright, so hello everyone and welcome to my session on biological approach. Um, Today, in this session, I'm mostly going to cover and review the theories that we've learned, like the common ones that we've learned, because um, I can't really cover the studies since every one of you guys, I'm sure, would have like the studies that you guys prefer to use. However, I do have like examples or suggestions, and if you guys want to ask me anything, feel free to ask. Um, so, oh yeah, since you guys can't talk, you guys are basically going to have to ask in the psychology channel, and I'll come back like every. I'll try to come back every time I finish like each topic. Okay. All right. So, like you guys know, these are going to be the things we cover: brain behavior, hormones and behavior, genetics and behavior, and animal research. Um, So we start with techniques to study the brain in relation to behavior. So there are different types of techniques that you use to study the brain, but usually in psychology nowadays we focus on brain imaging techniques, where you use technology to measure or image your brain structure, function, or the pharmacology of your nervous system. This allows researchers to see inside the living brain and know what's up, like when someone is conducting an observable behavior. So there are two common types of brain imaging techniques: structural, which shows you detailed images, and there's also functional, which lets you look at your brain functioning. And in this, we're going to cover. There's actually like a lot more than the ones that we're going to be covering. But we're just going to cover MRI and fMRI because they are two common ones. So MRI is magnetic resonance imaging. This machine, this big magnet basically produces 3D images. It allows you to look at tissues and organs very detailedly in black and white, like in this image, approximately like that. And basically what it does is that it has this big magnet that will create a magnetic field, and it will turn it on. It will be turned on when the person is inside the big machine, and then radio frequency will be pulsed to align the atomic nucleus in some direction. So in our body, we have a lot of atomic nucleus or protons, like hydrogen atoms, and basically they're going to like send like this imaginary waves to those, and then these nuclei are going to align to a certain direction. It's going to be turned on and off, so it's going to align and then go back to its original direction, and then align and then go back to its original direction. That is what you call pulses in a machine. And this like how these atomic nuclei behave is going to be detected by the scanner. So that way they can map the body, like the insides of your brain, by looking at how much energy it takes for one atomic nuclei to be aligned, and then how long does it take to go back to their original position, to equilibrium. These are like called radio wave signals, and they're going to be processed to form these detailed images. And then like some of the studies that uses these are Dragonski et al. 2004 and Megara et al. 2000. There's way a lot more than the studies here, like that you can use, but yeah, these are some examples. And then next we have functional magnetic resonance imaging, fMRI. This is like, as far as I know, this is like the upgraded version of fMRI, but instead of better image quality, they have like a way to look at the brain changes. So the way they do it is that they assume that when like your brain is active, your brain's oxygen supply would increase, so the oxygenated blood flow would increase. So they also use magnetic fields, but this time they try to see your blood level. So that's why they take the blood oxygen level dependent volt signal and try to analyze it to see the brain activity and to reconstruct it. So like let's say when they're doing something, they can do it while they're in the machine and measuring it, and they will be able to detect the blood changes that go straight in your brain. So it's an indirect way to see your brain activity. Alright, next we have localization. So localization is basically based on how specific brain parts are related to specific behavior. Since behaviors are complex, it would involve a lot of different brain parts. Specific brain parts has their own specific functions, and some of them are working together to create a certain behavior. And the common arguments that's around localization involves strict localization, which is how like a certain brain area would have that exact certain function. These are usually like highly supported in old studies such as the Broca-10 study. I'm not sure if you guys learned that, but it was a study where someone's like a certain brain area was damaged and this guy Tan couldn't speak. But that's not like a legit study for us to use in the psychology exam. But it's a good demonstration of strict localization. However, there's also several different arguments, because that argument is too stiff and basically like assumes all kinds of functions is localized to a certain part. So there was another proposed argument, which is that localization is distributed, like distributed evenly. I'm sorry, I think I forgot adding that up. So basically, it's the holism or the mass exponentiality, which is proposed by Carl Lashley. So the guy he tried to he studied the brains of the animals for years, and then he found out that apparently mice can retain their memory even after their brain is like removed, their brain portions are removed, but it becomes poorer in quality, and it doesn't matter which part of the brain is removed. And last, we have relative localization, which is that it acknowledges that some parts of the brain are localized, but it has limited. It also has some limitations to come with it. So, like for example, in H. Milner study, we know that there's this guy who got his hippocampus removed, and basically from there, researchers found that this HM patient couldn't remember anything, and they had severe undergrade amnesia, retrograde amnesia. And so he couldn't remember anything at all, like ever since he got his brain portion hippocampus removed. But he couldn't remember the memories before that occurred. And he also somehow, even though he forgot things, his motoric senses, when he on practicing, like doing the same experiment, his motoric senses improve. So you can see that like how memory is localized, like the creation of memory is localized in the hippocampus, but not necessarily all kinds of memories. There are different memories that might also be like linked with other brain parts, because you know how the brain is very complex. Okay, next we have neuroplasticity, which is the lifelong ability to reorganize your own brains, like the brain system, by forming neural connections through environmental events. So by adapting your brain. Basically, it can allow your brain to take a new function or redistribute the neurons that you have in your brain. And this effect is like the greatest in childhood. So before 12, this event is like exponentially increasing. It can grow really fast if you try to encourage its growth during childhood. Yeah. So some of the functions it has is it allows you to compensate people from getting injured or cortical remapping. So if like a certain brain part is injured, they can in the long term they can like your brain can reorganize itself in a way for you to still somehow live and adapt to life. So let's say like if you suffer your ability to see things because something happened in the back of your brain, then your brain is going to do cortical remapping, which is basically like they're going to find out how to use your other senses to be able to navigate around life. So basically, like they're not necessarily going to create a new function to like let you see, but they're going to use your existing functions and develop them to make sure that you're able to adapt to life. So, and then the next is to respond to environment change. This is usually more common in lab studies. Usually like with synaptic plasticity studies. So there are two different responses that they can make, which is neural pruning and long-term potentiation. So neural pruning is essentially when you have a lot of neurons in your brain and the brain thinks that you're not really using those neurons anymore, they will try to prune them, basically to remove them. So let's say when you're young, you studied ballet, so you have the ability to, be very, very flexible and remember a lot of ballet movements. But then as you grow up, it's no longer a relevant or necessary course, so you stop taking ballet courses. And then from there on, your brain will start forgetting, and your brain is going to think, oh, this part of your brain involving learning ballet courses are going to be useless in the future because you're not repeating it. You're not getting the same environmental stimuli, so they're going to neural prune those areas. They're going to prune them. And then long-term potentiation is basically just a fancy term to say if you learn a new skill and then they're trying to create more neurons by dendritic branching. So it's like what happens when you have an environmental stimuli and then have neurons fire continually in response to stimuli to create new dendrites and to increase the synapses available for behavior. So this is like to develop your neurons, but in the long term. So yeah, basically how neuroplasticity works is that you need to have some stimuli, and then this stimuli would affect your neurons to fire impulses, And these impulses will sprout new dendrites, and it will allow you to have increased synapses available for behavior and help you form a lot more neural networks. And if this stimulation doesn't occur, then the neural pruning would happen. If you guys don't really understand what's going on, I'm going to explain about neurons and their synapses more detailedly in the next topic, which is about neurotransmission. So basically, in neurotransmission, it's a chemical signal of neurons that transmit impulse across a synapse from one nerve to another. You may sometimes be thought of this as action potential, because apparently impulse and action potential is interchangeable, which is why sometimes it gets confusing when we talk about neurotransmission. We have different terms, a lot of terms. Okay, so as per the additional terms that we're going to be learning, this thing in the picture right there is going to be a neuron. This is a nerve cell that will be carrying impulse throughout the whole body, and it consists of the body, soma, this part, the areas with the nuclei looking kind of thing, or the eye kind of thing, the dendrites, which is what I was talking about before, the axon terminals. In the edge of the axon terminals, there's going to be tiny synapses, basically the axon terminals are usually going to be connected with other neurons through their dendrites, and between the gap of the axon terminals and those dendrites, they're called synapses, as seen in this picture. So as you see in this picture where it's zoomed, it shows you how neurotransmission works. Basically, the neurotransmitter is made in the soma, and then it's stored in the vesicles, in these circling areas, before it gets released into the synapse area. And then the impulse, which is an electric charge traveling to neuron membrane, appears, and it will release the vesicles. It would keep the vesicles open and move out the neurotransmitters to let the neurotransmitters bind to receptors in the next in the dendrite area. So this would transfer the impulse that was brought in from the original neuron to the next neuron through the synaptic cleft, and it's going to send that signal, and basically the neurotransmitter is going to be activated, or otherwise, if it doesn't get activated, or if it's done with their job, it would get transported back to the axon, or it would experience a reuptake. So neurons can excite or inhibit other neurons, turn on or turn them off. When neurotransmitters are sent to another neuron, they can become excitatory neurotransmitters, which creates the excite effect. So they turn the neurons on, and the inhibitory neurotransmitters, so they would create an inhibiting effect. Since there's, as you see before, like in this picture, you see that neurons have a lot of connections, right? And that means that it's not just one neurotransmitter sending a signal to another. So since there's also a lot of excitatory neurotransmitters and inhibitory neurotransmitters. So whether this effect actually occurs depends on whether you have more excitatory or inhibitory neurotransmitters. So if excitatory neurotransmitters is the majority, then it will turn on the neuron and like affect the neuron to get influence and make them want to also excite other neurons. That's kind of like how neurons communicate with each other. And with enough stimulation like this, they would be able to produce behavior. So that's why neurotransmission's behavior production increases by chance. So it's like an indirect effect, but it increases the chances for it to occur. And then neurotransmitters can also be affected by agonists and antagonists. An agonist is a chemical that enhances these neurotransmitters, while antagonists are chemicals that counteract these neurotransmitters. So examples of agonists would be, let's just say in antiviral, we use I don't know how to say it. So basically we use acetylcholine as a neurotransmitter, and we also have its artificial drug which inhibits the acetylcholine neurotransmitter. So the agonist would be the acetylcholine, and the antagonist would be the drug that inhibits the acetylcholine effect. And in neurotransmission, there's a lot of neurotransmitters, right? There's serotonin, there's acetylcholine, there's dopamine, adrenaline, and there's a lot of different studies that shows its effect. So I only have 2 example studies here, and I'm pretty sure you guys have more that's recommended by your teacher, and I'm pretty sure that's fine. Okay, so do you guys have any questions? Oh yeah, I forgot to send the slides. Let me send the slides for a second. Also, I didn't say Andrew Reynolds. What is Andrew Reynolds? I'm sorry, but I don't know that person. Who's that? What? Oh wait, is it that? Okay, that's the biological approach slide, guys. Thank you, Nat. And wait, no, it's Alex. Thank you, Alex. Is there no questions at all? Yeah, I didn't say Andrew Reynolds. I just did just now, but I didn't say it before. Sure, go ahead, ask a question. Yeah, unfortunately, I think you guys can't speak anymore. Wait, I forgot to change the permissions actually. Okay. In hormones, they use adrenaline as a hormone. So basically, for some of these, they can function both as a hormone and a neurotransmitter. So whether it's really hormone or neurotransmitter, I'd say it really depends on the study that you're using. Because like neurotransmitters and hormones sort of overlap with each other. One is like in the brain, and the other is in the bloodstream, but they're kind of in some way related to each other. I think I have a slight explanation on that later on in the hormones and behavior section. Oh yeah, and by the way, the concepts that I'm explaining is like kind of a lot more too elaborate than your usual psychology explanations. So don't worry if like your explanations aren't as complete as this. Yeah, because I just need to understand what's going on. I just like enjoy trying to understand what I'm studying instead of blindly memorizing them. Okay, so I suppose we can proceed because I don't really see any more questions. Alright, I guess I'll proceed with the explanation of hormones and behavior then. Okay, so hormones are basically biochemical messengers. They usually regulate the long-term functions of our human body physiologically, like growth, metabolism, digestion, and reproduction. They are also released by the endocrine system and hard to fully control because it's your long-term bodily processes. They're also connected with neurotransmitters but independent. And they can also interact with each other. So the reason why they're connected somehow is because even though they're released in the endocrine system, there's this one section in the brain, I think called the hypothalamus, which also functions or has a link to like your hormones. And sometimes for some hormones, like oxytocin, they're produced in that hypothalamus. So that's why they are somehow connected with neurotransmitters. Hormones also only influence cells that have receptors for hormones. So for the cells in our body that is a target cell for the hormone, they can influence. And the way hormones work is that in that like target cell, they bind to the receptor, and this binding causes your genes to change. So it can activate or suppress our genes. Thus, they don't directly influence behavior, but increases the chances of the behavior happening. And there are different types of hormones that you can encounter in like studying psychology and the studies that you use. I think the two common ones that I've encountered is oxytocin and cortisol. And I'm pretty sure there are the ones for adrenaline too. So, yeah. I mean, for these, I think it's like you just need to know about the common knowledge, and there's not a lot of things I can talk about for this part. And next, we have pheromones, a chemical messenger. This time it's produced and released in an environment to communicate information from one to another, and it's common to both genders. For animals, it has two types, which is for long-term physiological changes like hormones, or for rapid behavioral effects, such as mating. It involves attraction, following trail of another animal, parent-child interaction, and etc. It's innate, not learned. So it's a biological thing, basically. So pheromones is very interesting, like in humans, because up till now there's no specific pheromones identified. However, there are a lot of putative ones or potentially hypothesized pheromones that are being discussed, being studied, and a lot of the results are inconclusive. However, the IB includes this so that we can practice our critical thinking and evaluation when we talk about pheromones. So, basically, in animals, they process the pulmononasal organ. I'm sorry. I think I was like trying to move the abbreviation up and I forgot to include it back. Yeah, so animals process the pulmononasal organ through. Animals process through the pulmononasal organ in the nasal cavity. So this pulmononasal organ, from nose, nasal, this is connected to the AOB, the accessory olfactory bulb. Basically, this is a name of a brain region. This brain region is adjacent to an olfactory bulb and a region responsible for processing regular smell. So in animals, their pheromones has something to do with smell. However, it's not something that you can immediately smell right away. It's something that can be picked up by the nose, but it doesn't have a scent. Like, it doesn't have a particular distinct scent to it. Meanwhile, in humans, they have different physiological structure. So, like, for fetuses, for babies that are not born yet, they have accessory olfactory bulbs. However, upon birth, they disappear for some reason. And then not every human have pulmononasal organs. And it's not connected to the nervous system. So it seems like humans' biological foundation for pheromones is kind of different from animals. But it doesn't discount that the pheromone system exists. Because it's just a matter of how we're like biologically wired differently. So, and the reason why there may be a putative one, but not necessarily like the same as animals, is because humans have like around 400 oral receptors with different genetic variants. So, there may be with this much different odor receptors, it might be that there's a certain odor or a certain gene that might be detected, that might be associated by smell and detected, and communicate and does an indirect signaling similar to in animals. And some of the studies that you can use includes Medigan et al., which investigated about the major histocompatibility complex, MHC genes. Lundstrom and Olson, which investigates about the androstatinol, and also inheritable, and there's a lot more too, actually. Okay. Now we're already in genetics and behavior. Do you guys have any more questions? Okay. No questions at all? Okay, if you guys have any questions, you guys can just drop it in the chat. And also, I'm not sure if you guys can already talk, but I already removed the terms for I already added the unmute terms, so you guys can already talk if you guys want to. Yeah. Okay, if no one has any questions, I'll just move on to the next part. Next, we have genetics. Genetics is essentially a unit of DNA located in a chromosome. It's the most basic and functional unit of heredity, which controls the development of one or more human characteristics. So it's like one of the foundations of passing down information from one parent, one human to another human, which is from parent to offspring. And usually, this entire genetic mapping or genome, it acts as a blueprint of our structure and function of the human body. But we're still studying this. At least the scientists that are studying genetics are still studying this and still aren't able to fully map the entire human body's genetic sequences, genomic sequences, and understand each of their functions very well. So there's genotype and phenotype. Genotype is basically a specific set of variants of gene in a person's DNA, while for phenotype, it's the individual characteristics like trait, appearance, and behavior. Genotype, the gene name, and then phenotype is basically the thing that you can physically or behaviorally observe. And next we have basically the genes are usually responsible for phenotype expression. So one gene won't necessarily be responsible for exactly one phenotype. However, it increases the likelihood for a phenotype to appear. Especially in behavior. They don't just let's just say the aggression gene, MAOA, the gene that is associated with aggression. That MAOA gene having that gene with a specific mutation doesn't mean that you will have aggression, but it just increases your likelihood of being aggressive depending on your environment. So your environment also still plays a role. That's why research implicated the functions of specific genes in various behaviors. And next we have genetic similarities, which is how humans share genetic similarities, especially with family members. The monozygotic twins or identical twins share 100% of their genotype. The offsprings from parents usually share 50%, while siblings and dizygotic non-identical twins share around 50%. These similarities tries to look at what extent gene affects behavior in order to study this entire realm of genetics, because it's, as explained before, it's hard to look at people's genetics and its effects. And whether a behavior is actually caused by genetics or caused by environmental effects. So there are different studies that that researchers undertake, which are like notable enough to be discussed about, which is twin studies. One of the most common like genetic similarity studies that people use. If twins behave similarly, then there might be a chance that genes plays a role. However, there's also the consideration of environmental factors. So in different cases, sometimes researchers would do adoption studies instead between twins or between siblings, because it allows the test to see if the environment affects people's cognitive abilities. Let's just say an example would be someone's IQ. That's one of the most common tests that people do. Or they can also investigate about depression. There are, for adoption studies, there are two different angles they can look at this problem. One is the correlation between children and the parents, the adopted children and the foster parent, and then comparing it with the biological children and the biological parent. There's also comparing between different siblings without biological parents. So if you have two twins and then both of them has their own families, you can compare their cognitive abilities. Okay, so I thought this was like worth discuss because this is taken from the Oxford book, and they like offered really interesting limitations that you can take from when you're writing your ERQ. And some of these are regarding twins and adoption. So the first is how usually twins have a small sample size and are rare. So there's less replication studies and less opportunities for researchers to look into these studies. In addition, since twins is like a special population, they're not really like the same from regular people. That means that there's a chance that they're not very representative of the population, because they may have their own special characteristics and differences from regular people that we can detect, like something that occurs during the pregnancy stage that can affect them. Furthermore, similar physical features can also affect similar results. So if both of the twins are both attractive, let's say like both physically attractive, then they would tend to be treated better than any people. So their environmental influence would still be quite similar in the end. So it's going to be hard to kind of distinguish between whether it's actually a gene thing or if it's an environmental thing. Finally, having twins, even identical twins that are reared apart or adopted with like different families, or one biological, one another family, is limited, because most of the time when these adoption studies occur, there's the prenatal environmental factor, aka the factor where twins are together until like a certain point of time, and the time when they're together is significant enough for them to be similar to experience significant similar environment factor. Thus, coming to the next point of not directly separated at birth. Because usually, like, if twins are born, you don't just separate them right away. Like, put them literally in different rooms and stuff. And even if they were, the people that adopt them or the places that they are adopted in tend to have similar cultures and socio-economic backgrounds. So it's still going to be hard to distinguish between the gene and the environment in that context. Okay, so some of the studies that you can use include Bosha and Mugoye, Canada et al. 2015, and there's also a lot more that you can use. I expect that you guys would already have studies that you guys are going to use. However, if you guys don't, you can always check in the helper website, because there's a lot of study notes that people have made for these. So next, we have evolutionary explanations. Evolution is something that changes over time, depending on the heritability of the characteristics of a species. Sorry, let me rephrase that. So evolution is something like the heritable characteristic of a species that changes over time, depending on the environment, especially in order for people, at least for creatures, to survive. This is done through natural selection. So some of the assumptions that people make about evolution is that organisms, such as us humans, are different to survive and reproduce. And to continue that, they pass down genes that are good enough for us to survive. And this explains the variety of species and allows us to trace humans and different animals as a common ancestor. This allows for animal research opportunities. Okay. And however, evolutionary explanations are also met with some criticisms with the theories that are proposed. The first one is the speculations on the environment. So there's very scarce knowledge on environmental adaptation that occurred thousands of years ago, because we don't have scientific testing and human records that are from thousands of years ago. So a lot of the things that are discussed in the realm of evolutionary explanations a long time ago is speculated or what ifs, hypothetical situations. So they're not very reliable. Furthermore, it's very difficult to test the behavior in a long term like this, and tend to have an ad hoc fallacy. Basically, what's happening is that it tends to take a phenomenon and assume that it's true. In the case of evolutionary explanation, they often take the argument of how humans are hardwired to survive, so they develop a certain behavior. That argument tends to be assumed to be true without testing if it's true or not. However, there are studies, if I recall correctly, there are studies about disgust. I don't remember the author, but I can share it to you guys later if you guys want. They try to test out the assumptions by creating a certain logics onto it. So if they manage to test this correctly, then the assumption is true. So they try to rule that part of the criticism out. Furthermore, evolutionary explanations also tend to assume that a trait has been gradually evolving. So it's constantly at its peak and trying its best to be the best genetic trait. And this is not always necessarily true, because evolution is not like a straight path. Sometimes it may be that it used to have a certain function, but then that function sort of just disappears or gets used for like a different function. An example of this is acceptation, which is like an example of how birds used to use feathers for temperature regulation. But through evolution, they managed to use their bird feathers to also be able to fly. And next, we also have the lack of cultural variation. So nowadays, with like a lot of different ethnicities and race, we have a lot of different cultural variations, right? And unfortunately, with evolutionary explanation, it's very hard to account for those for those cultural variations, because the thing with cultural variations is that it's very hard to like scientifically track and assess. So only universal traits that are consistent throughout all the human species. One of the obvious ones includes disgust or mating attraction. Only those can be tested. And finally, it's also hard to differentiate between adaptation or neutral processes. So there are times when the gene would mutate and evolve. However, it's not evolving for the best cause, but it's just mutating because it's a random process that naturally happens. That's usually very hard to differentiate in evolutionary explanation. However, evolutionary explanation is very interesting in a way that this may be the argument that can tie up all the different biological like explanations or social cultural explanations or cognitive explanations of human behavior, because it tries to explain things in the big picture. The thing with like the biological explanations that I explained before, and with the social cultural explanations that I will be discussing in my next few sessions, is that they tend to be reductionist. So they only focus on that specific aspect, and it's hard for them to like integrate and tie it to different approaches. So evolutionary explanation can be that one thing that ties them all together. Okay, so we finished SL. We just finished SL, and wait, do you guys have some questions before we move on to HL? Oh yeah, by the way, this means that the SL kits are done. Yay! So you guys can leave now if you want to for SL. Oh, you guys are also wholesome. Mohan, please sleep. Okay. There's actually a little bit more for animal research, so I'll just cover it real quick since it's only been an hour. That feels so weird. Okay, so I'll just like cover animal models really quick. Okay, so For animal research, you have value of animal models and providing insight to human behavior, right? And the topic of value of animal models is kind of like overlapping and similar to providing insight to human behavior. Like they have the same main point. The thing with them, both of them, is that you're going to have to frame your discussions differently later on in your ERQ. Okay, so animal models. Animal models uses animal research, like non-human species, to understand human behaviors better in the physiological perspective. So they try to compare biological stuff and observe animal behaviors and try to generalize them to humans. The reason why they can do this is that there are certain species, animal species, that are quite similar to humans. So, let's say for primates, they have an average of 96% similarities, with the most identical as chimpanzees and bonobos at 99%. Or rats and mice are often at around 95%. So that's quite good for generalizing to human characteristics or attributes. Historically, humans actually played a very important role in studying human anatomy and human physiology because it provided lots of valuable contributions in the foundation of physiology, neuropsychology basis of hunger, thirst, and sexual motivation. So a lot of those people the stuff that people say are human instinctive drives, yeah, those kind of things. So there are different advantages and limitations that comes with evaluating animal research, the use of it. One of them includes how humans and animals' biological state and behavior are similar. So, since we have quite similar genes with certain species, human animal species, we can study them in animals and get similar results in humans. Some of these includes acetylcholine and its effect on spatial memory, or neuroplasticity. Neuroplasticity, because in animals, neuroplasticity can occur, and that's how they also sort of discovered that it's also possible to promote synaptic plasticity in humans. Sorry, yeah. It also allows you to investigate animals on their entire lifespan. So, you know that humans tend to investigate an entire lifespan of humans, you need to live for an average of 60 to 70 years. However, for animals, you can just study them for 5 years or even less than that for some animals. So it's easy to study certain lifespans with that. Or if you want to study the entire lifespan. Furthermore, there are a lot of physiological variables that can be manipulated that are very ethical in humans, such as inbred selection. So selecting animals from the same parent or the same from the same bird, because for some animals they can give birth to several animals at the same time. So let's just say for rats, they can take the animals that are from the same litter. And then they can also control the environments in the animals to make sure that they are exactly the same for each animal. The ultimate control that we can't have in humans. Furthermore, it's also unethical for humans. Yeah, these kind of things are very unethical for humans, and also allows us to study like very detailed anatomy related things that we can't do in humans. However, similar doesn't mean that they're same. There are some generalizability issues that occur when studying between animals and humans, which is especially prominent in the psychological aspect or in like cognitive or social cultural approaches. Usually psychology is more well known in like its cognitive and social cultural like approaches. Like theory wise, it's more about like the mind, personality, or environmental influences, not so much with biological stuff. I mean, even if it's biological stuff, they tend to also directly study humans, since you're going to have to test it again in humans. And that's why it seems like animal research is not that important in psychology, but to some extent, when you're studying a new like biological related phenomenon, like very biology and behavior related, animal research is still very important as like the baseline of the foundation of the research. Furthermore, animals can also behave differently to humans, so that's why studies on humans are still needed. And furthermore, the differences in humans and animals is that we have more advanced executive functioning or like cognitive abilities. Most animals, I think, can think of things in abstract things, like what we're doing right now. What we're doing is like thinking about theories, thinking about phenomenons that are occurring like in a descriptive way. This is considered like executive functioning, and most animals can't do something like this. Furthermore, animals can't communicate responses to us humans, so we can only observe them. So it like the observation in animals and humans are also limited in different ways. And also, because animals are organisms and they also have lives and they also have their own consciousness, ethical considerations is still important and needed for animals. Thus, there's the 3R for animal consideration, which is to reduce, refine, and replace. Reduce is to reduce as much the use of animals if possible. Like, try to take the minimum number of animals that you can use and use it effectively as possible. And it's also important to reduce human suffering and give them like appropriate food, clean environment, etc. Because it's not nice to torture and kill another organism, even though we can't communicate with them. Furthermore, it's also important to replace or use alternative techniques. So, like for example, if you can do the if it's still possible to do the experiment on humans, then opt for human experiments instead of animal experiments, or use alternate techniques like computer simulations, using animals in the lower food chain, like using rats instead of dogs, and etc. And that concludes our session. So this is just like some of the suggestions for the studies that you can use for the HL extension. Thank you for attending my session, and good luck for whoever is taking your exams. Thank you for coming to my session.